my absolute honor and pleasure to introduce Dr. Helen Collins, who's an assistant professor in the Department of Medicine in the Center for Cardiometabolic Science, and she's associate director of the Diabetes and Obesity Center. As you will hear from her accent, Dr. Collins is originally from the United Kingdom. She received her bachelor's with honors from the University of Leicester, and her interest in cardiovascular research really began as an undergraduate and has just continued on over her career. She did a Ph.D. in cardiovascular sciences, also at the University of Leicester. And that work resulted in research papers published in Circulation Research and in the Journal of Hypertension. She then was a research assistant at the Open University, and then was a postdoctoral scholar in the Department of Pathology at the University of Alabama at Birmingham, where she published 14 papers, including 2 papers that were part of an ongoing collaboration with one of her postdoctoral mentors, Dr. Chatham. She joined U of L in 2019, and she has really thrived here as part of the cardiometabolic science group headed up by Steve Jones. She's participated in a number of training opportunities in teaching and mentoring, and she has received a number of awards, including the 2022 Division of Environmental Medicine Faculty of the Year Award here at U of L. She's on the editorial boards for 8 journals, including Molecular and Cellular Endocrinology. So thank you for saying yes to that. And she served on innumerable grant review panels. So she was named the Star Reviewer for 2021 by the American Journal of Physiology, Heart and Circulatory Physiology. She's currently serving on 2 Ph.D. committees and mentoring 1 master's student, and has trained 2 NIDDK T35 summer trainees, and we'll have 1 again this summer. She's currently PI on 2 NIH Heart and Lung HL R01 grants, and she's co-investigator on another R01 grant. And she's published 24 papers with many studies in progress. She's been an invited speaker to a number of national and international meetings, and she has an upcoming presentation in September in Seoul, South Korea at the International Congress on Lipid and Atherosclerosis. So that sounds like it'll be a fun trip. Well, thank you for speaking with this group today.
Thank you, Carrie, for that kind introduction. Well, thank you for that kind introduction. And I'd just like to take a minute just to draw attention to the title of my talk. In no way am I making light of the issue of the maternal mortality rates in the US. But what I want to propose is that we all want every woman in the US to have a smooth pregnancy. But at the moment, we are actually wondering, Mummy, are you okay? Are you okay? So other than these specific funding disclosures, I have no additional disclosures. So pregnancy in the United States. Recently, in the past several months, a study was published in which they specifically noted that potentially there's issues with the documentation and reporting of maternal mortality rates in the US. And this caused a lot of concern, and many of the research agencies significantly disagreed with the findings of this study due to some flawed methodologies within, and this significant undercounting of maternal deaths, which were contained within this body of research. And this was largely condemned by the CDC and the American College of Obstetricians and Gynecologists. So if you take away but one thing from this talk, please take away that actually pregnancy in the United States is dangerous. This scheme here highlights the maternal mortality rates in the US for the year of 2018. You'll see that the US leads the pack with 17.4 maternal deaths per 100,000 live births. And this rate is much higher than the rest of the developed and developing world. And more recently in 2020, these stats were updated. And now there's been a significant increase between 2018 and 2020, such that there are 23.8 deaths per 100,000 live births. And you can only imagine that with the change between 2020 and 2024 today, that this number has only increased. If we take a look at a state-by-state basis with respect to maternal mortality rates, you'll notice this data, well, first you'll notice that it's slightly outdated because it's from 2005 to 2014. But what you should appreciate is that during that specific time period, many of the states in the US had significantly high maternal mortality rates, indicated by the intensity of this pink color noted. You'll notice that the high intensities of the pink color are concentrated mostly in the southernmost states of the US, but you can only imagine how this map has changed since 2014. And here's Kentucky in 2014, there were 13.5 maternal deaths per 100,000 live births. But if we go back to that original starting slide where the national average was documented in 2018 at 17.4 deaths per 100,000 live births, at this very same time, you'll appreciate not only has there been an increase in the national average between 2014 and 2018, but the Kentucky average significantly increased from 13.4 deaths per 100,000 live births to 76 deaths per 100,000 live births. So this is a significant increase in maternal mortality in Kentucky. And what is abysmal and disgusting about this is that it was shown in the same cohort that 91% of maternal death in Kentucky was preventable. So we need to do a better job at reducing maternal mortality rates, not only at the state level, but also the national level. So I'd be remiss not to discuss the significant racial disparities that exist with maternal mortality rates in the US. You'll note here from this scheme that Black non-Hispanic women have significantly increased maternal mortality rates in comparison to white non-Hispanic and Hispanic women. If we go to look at the states with the highest maternal mortality rates, you'll actually see that these states have also the highest shares of Black births. So tackling racial disparities with respect to maternal mortality rates is a significant issue that we have to tackle within the coming years. So what contributes to some of these maternal mortality rates? So it has been shown that cardiovascular disease is the leading cause of maternal mortality. If we look at this graph here, you'll note that 15% of all pregnancy-related deaths are comprised of cardiovascular diseases. If we expand this further to look at the specific cardiovascular diseases that occur and impact pregnancy, you'll note that this is fairly widespread, and we see that there's significant contributions from arrhythmias, coronary artery disease, cardiomyopathy, congenital heart disease, valvular disease, and then this other 4% here is comprised of several pregnancy-associated complications that occur in the later stages of pregnancy, such as preeclampsia and peripartum cardiomyopathy. It's important to note that even with a normal pregnancy, there are several cardiovascular symptoms that indicate normal physiological changes, such as breathlessness upon exertion, fluid retention, and increases in heart rate and intermittent palpitations. But these are all usual symptoms and signs of a normal pregnancy. We need to increase awareness of cardiovascular clinical symptoms within women, and this is especially important across the reproductive lifespan because many women are just simply not aware of the cardiovascular clinical symptoms which can impact them. And this is a direct result of the lack of research that has been done within female populations and the lack of awareness of cardiovascular clinical symptoms amongst women. So it's important to note that these symptoms do occur, but when they become extreme, such as we see extreme breathlessness, chest pain, tachycardia, that's when we need to be aware and seek attention. So several maternal cardiovascular disease factors have arisen over the years. Within the last decade, advanced maternal age has arisen as a significant risk factor for maternal cardiovascular disease, with many women choosing to have children later in life. Also, in addition to this, multiple pregnancies, such as changes with the gravidity, which is the number of times a woman has been pregnant, or parity, the number of times a woman has given birth, and also multiple pregnancies such as twin and beyond. Also, coming from the Division of Environmental Medicine, I'd be remiss not to mention that smoking, e-cigarette use, and environmental exposures to toxicants and pollutants are significant contributing factors for maternal cardiovascular disease. In addition, as previously mentioned, preeclampsia and hypertension, obesity, poor nutrition, and pre-existing or gestational diabetes are known to precipitate maternal cardiovascular disease risk. In addition, you may be surprised to know that the normal process of a cesarean section imposes some maternal cardiovascular disease risk. In addition to this, the normal process of lactation also contributes to maternal cardiovascular disease risk, and this is most highly notable within the ailment of peripartum cardiomyopathy, where you get inappropriate cleavage of the lactation hormone prolactin, which results in pathology. In addition, infection and autoimmunity are also significant risk factors. So as I alluded to on the earlier slides, we need to do a better job of increasing awareness amongst women across the lifespan of the cardiovascular diseases that can occur during pregnancy, but also increase awareness amongst healthcare providers and the general public. This will go some way to then promote women in joining several clinical studies, because at present, many women in general, and specifically pregnant women, do not usually enter clinical trials. So we know less information about them. So increasing awareness will also help promote inclusion in scientific research. Also, we need to improve access to healthcare and improve existing pre and postnatal care. You might be surprised to know that a recent study by the March of Dimes suggested that around 80% of US women do not have appropriate access to good maternal health care. So increasing access to health care is important, as well as improving the existing healthcare measures that we have. Also, the consideration of chronic health conditions is important. Some women go into pregnancy with pre-existing cardiovascular disease, so it's appropriate to consider this and manage those pregnancies appropriately. And then, as I mentioned, we need to tackle racial and socioeconomic disparities. And then, if women know of the cardiovascular symptoms early, they can seek timely intervention. And then also, if we develop a panel of effective biomarkers, then we can detect at-risk populations of women. Why this is important is because maternal cardiovascular disease has a long-lasting impact on female cardiovascular health. This recent study by FAHA, which took women under the age of 25, between the ages of 25 and 35, as depicted here, and those older than 35, and at each age, the presence of a cardiovascular complication during pregnancy contributed to increases in all-cause mortality and cardiovascular mortality. So decreasing maternal cardiovascular disease will also decrease female cardiovascular disease in the long term. But in order to us to fully appreciate this, we have to have an understanding of the changes that occur with a normal, uncomplicated pregnancy. So what are those changes? So in the cardiovascular system, significant changes occur to meet the maternal fetal circulatory demands. These include increases in cardiac output, mediated through increases in heart rate and stroke volume. There's also reductions in total peripheral resistance, changes in blood pressure, and enlargement of the heart. This enlargement of the heart is a physiological hypertrophy that occurs during pregnancy. And this is notably different to what is observed during pathological hypertrophy, such as what would be seen with a transaortic constriction or heart failure induced by MI. So with a pregnancy-induced hypertrophy, we see a preservation of cardiac function. This hypertrophy is reversible. We have minimal fetal gene induction, and we have no fibrosis. And although we have several signaling pathways which contribute to both pathological and physiological hypertrophy, these types of hypertrophy differ substantially. So there have been several proposed mechanisms to contribute to pregnancy-induced cardiac growth. These include the myriad of hormonal changes that occur with each trimester of pregnancy, and these hormones are known to directly impact cardiovascular physiology. Also, the mechanical stress imposed on the heart, such as through the release of growth factors and cytokines, also are thought to contribute. In addition, the increased workload imposed on the heart through hypervolemia and the need for the increase in cardiac output to meet the circulatory demands. And also additional factors such as nutrition and metabolism, which I'll discuss in subsequent slides, are thought to also contribute. However, despite some of these proposed mechanisms, the specific mechanisms that contribute are not well understood. So in order to tackle this head on, in my lab, we have tried to recapitulate some of these pregnancy-associated changes in our hands. So in order to do this, we've taken 12-week-old FEB and J mice, and for the most part, examined them in the rest of this presentation using these 4 main time points. We have a non-pregnant group, a mid-pregnant group, which reflects day 8 of pregnancy, a late pregnant group, which reflects day 16 of pregnancy, and a 1-week post-birth group where dams are allowed to maintain their litters. So not surprisingly, we see during pregnancy, there's an increase in body weight, which peaks at late pregnancy, which is the point of maximal fetal growth. We also observe increases in heart weight and heart weight to tibia length at all stages of pregnancy and post-birth. Also, in line with the predefined criteria for physiological hypertrophy, we do not observe any changes in cardiac function, and this was assessed through echocardiographic measurements of ejection fraction. We don't see any changes at any time point during pregnancy and post-birth, but what we do see is increases in chamber dimensions of the heart, such that we see an increase in left ventricular end diastolic and end systolic volumes. And also we see an increase in cardiac output, which in our studies is primarily mediated through an increase in stroke volume rather than heart rate. Also in line with this criteria, we observe no appreciable fetal gene induction as evidenced from the gene expression data here of MPP-A, MPP-B, and myosin heavy chain 7. In addition, we do not observe any evidence of cardiac fibrosis in the maternal heart as determined through picoserous red staining, where we see no changes in percent fibrosis at any time point. So having standardized these hypertrophic changes in our hands, we next wondered what the specific molecular mechanisms contributing to pregnancy-induced cardiac growth were. So it's known that during pregnancy, there are several significant changes in systemic metabolism and cardiovascular metabolism. We see increases in circulating metabolites such as fatty acids, triglycerides, and ketone bodies. But also at the level of the heart, we see reductions in glucose metabolism, increases in fatty acids, and increases in ketone body metabolism. So recently, a study from Zoltana Rainey's group assessed the late pregnant heart and found that the pyruvate dehydrogenase kinase 4, which is an inhibitor of pyruvate dehydrogenase, was significantly increased in late pregnant hearts. This was also associated with an increase in phosphorylation of PDH, and through subsequent studies in the same publication, they found that this increase in PDK4 was mediated primarily through an increase in the pregnancy hormone progesterone. But most excitingly, they found that when you inhibited PDK4 with dichloroacetate, this in fact inhibited cardiac growth and was suggestive that a reduction in glucose oxidation is important for cardiac growth. So with these metabolic changes in mind, we next thought to examine the cardiac transcriptome, metabolome, and proteome of the maternal heart at various stages of pregnancy and post-birth. So I'll briefly discuss the changes at mid-pregnancy and post-birth, but I'm going to focus on those mainly during late pregnancy. So what we observe in the mid-pregnant heart is increases in transcripts associated with angiogenesis, cell proliferation, and primarily the vascular endothelial cell compartment. At post-birth, we also see increases in transcripts associated with the extracellular matrix and also fibroblasts. But at the late pregnancy time point, we see an increase in transcripts associated with metabolism. We also corroborate the Irani lab findings, whereby we see an increase in PDK4 expression, in addition to several other metabolic transcripts here, including oxyglutarate dehydrogenase. So in order to confirm these changes at the protein level, we next performed immunoblot studies. And indeed, we were able to confirm this increase in PDK4 expression at the protein level, where we saw an increase in PDK4 expression at late pregnancy. We also observed a reduction in the phosphorylation and thereby activity of PFK2 in late pregnant hearts. And although I didn't allude to it at the time, when we performed the transcriptomics analyses, we also found that the ketone body enzyme beta-hydroxybutyrate dehydrogenase 1, or BDH1, was significantly increased in hearts at all stages of pregnancy and post-birth. So we also then validated those changes at the protein level, and we were able to recapitulate the changes that we have observed in the transcript at the protein level, in that BDH1 expression was increased at all stages of pregnancy and post-birth. So what we believe is happening is that we are seeing a reduction in glucose oxidation at the level of both PFK and PDH. And this is mediating redirection of glucose-derived carbons into these pathways here, which have been previously associated with pro-growth or anabolic pathways, which are pathways which provide the building blocks necessary to facilitate cardiac growth. And at the same time as that's occurring, we also believe that metabolism is maintained through ketone bodies and BDH1. So in order to try to ascertain whether this is in fact the case, we next perform bulk metabolomics to see if any of these ancillary pathways of glucose metabolism, whether any of the metabolites associated with these are changing the maternal heart. I first like to bring your attention to the fact that we see minimal changes in the cardiac metabolome between non-pregnant and mid-pregnant hearts. We see the bulk of the changes in the cardiac metabolome between late pregnant and post-birth hearts, which are the major time points associated with the cardiac growth that we observe. So we see specifically at late pregnancy, with respect to the metabolome, that several metabolites associated with amino acid metabolism are increased in abundance. Specifically, we note that the liver-derived urea cycle-associated metabolite homoarginine is significantly increased in late pregnant hearts, in addition to several other amino acid metabolites. We also see increases in fatty acid metabolites, specifically sphingomyelins and long-chain fatty acids. Moving along to the post-birth time point, we also see, once again, amino acid metabolites increased in post-birth hearts. Once again, we see urea cycle increased, but now we also see polyamine metabolites increased, and they have already been associated with hypertrophy. We also see the nucleotide metabolites, such as pyrimidine and purine metabolites, are significantly increased in abundance. In addition, we see that glycerophospholipid metabolites are also increased. So this is supportive of our theory that there's increased, potentially increased flux and increased direction of glucose towards ancillary biosynthetic pathways associated with cardiac growth. But the caveat to this is bulk metabolomics. It only gives us a snapshot of what's happening in time. So we wanted to know if flux into these pathways was significantly increased alongside this. So we then performed stable isotope resolved metabolomics. And this study where we have, once again, our 4 main time points, and these mice are fed a liquid diet with regular glucose, and then after a period of acclimatization, they are switched to a liquid diet containing 13 C labeled glucose for 18 hours, which we've shown to be sufficient to label the ancillary biosynthetic pathways of glucose metabolism. And what we see in support of our bulk metabolomics is we see increased incorporation of glucose-derived carbons into these pathways associated with cardiac growth, such as nucleotide metabolism, membrane expansion, which would reflect glycerophospholipid metabolism, and amino acid metabolism. In these experiments, we also included another group, which we refer to as post-birth non-lactation. And this is essentially a matched group for this 1 week post-birth group. However, the difference is within as fast as possible after birth, the pups are removed from the dam. And the reason we did this is because we found that lactation has a profound effect on pregnancy-induced cardiac remodeling. So we see that following removal of the pups, there's a significant regression in cardiac size to levels which are comparable with late pregnant mice. So we propose that, given that we see significant increases in incorporation of glucose-derived carbons into these ancillary biosynthetic pathways to promote growth, that maybe we might see the inverse of this in this post-birth non-lactation group. And indeed, that this is the case. We see that several of these flux into these metabolite pathways, which we had previously noted, is significantly reduced in the post-birth non-lactation groups. In addition, during these specific studies, we also, as I mentioned, assessed the cardiac proteome at various stages. And so once again, we note that there's minimal changes in the cardiac proteome in going between non-pregnant and mid-pregnant hearts. We see the bulk of the changes in the cardiac proteome in the late pregnant and post-birth groups, which are the groups associated with the significant cardiac growth. Specifically in these two groups, we see lower expression of mitochondrial complex subunit proteins, specifically those associated with cytochrome C oxidase and NADH ubiquinone oxidoreductase. In addition, we see decreases in several histones and contractile proteins. So in order to ascertain the physiological relevance of this decrease in mitochondrial complex subunits, and to also garner some information with regard to substrate use in the maternal heart, we next went on to examine cardiac mitochondrial function in pregnancy and postpartum. We once again have our 4 main time points or groups to include. We performed gravimetric analyses, and then we isolated cardiac mitochondria. And the primary endpoint of this study was to examine mitochondrial respiration using seahorse technology in response to several different substrates. First being pyruvate malate, which represents a carbohydrate source, octanyl carnitine malate, which represents a fatty acid source, also glutamate malate, which represents an amino acid source. So that was our primary endpoint, but we also saved back any isolated mitochondria that we didn't use for these studies for immunoblotting. So what we first show with our gravimetric analyses in these studies is that we recapitulate all of our previous findings in that we see increases in body weight, which peak at late pregnancy, such as that we've seen before. And we also see that characteristic hypertrophic response where we see increases in cardiac size throughout all stages of pregnancy and post-birth. What we don't see, however, is any specific change in crude mitochondrial yield at any time point of pregnancy and post-birth. Also, within this study, we went back to reanalyze our cardiac transcriptomics and proteomics data sets. This is one because we, on the first pass analysis for RNA-seq especially, usually mitochondrial encoded transcripts and ribosomal proteins are excluded from analyses. So we redid the analyses of those, included the mitochondrial encoded transcripts and ribosomal proteins, but we also aligned it to the most recent version of the mitochondrial genome. And in those studies, not only did we recapitulate the specific changes that we had noted with the bulk transcriptomics with respect to PDK4, BDH, but we also saw that several solute transporters were changed in expression. And also we observed that potentially some mitochondrial fission and fusion proteins were changed, but this data here suggests that potentially this may not have an impact on our studies here, but future studies will interrogate this in more detail to actually examine mitochondrial ultrastructure in the maternal heart and postpartum heart. But getting into the body of the mitochondrial respiration studies, so we had recapitulated what we had found. We had found that there's no change in mitochondrial yield. Next, we performed the seahorse analyses. And so this is our carbohydrate source of pyruvate malate. We see an increase in state-free respiration in post-birth mitochondria. And this was also, we also observed an increase in RCR at post-birth, which suggests that post-birth mitochondria are able to respire on carbohydrate sources, and this is efficient. We also see with the amino acid source of glutamate malate, we see that post-birth mitochondria have increased state-free respiration compared to the other groups. And this also, similar to the pyruvate malate data, results in an increase in efficiency with the provision of this substrate. So because we had previously shown that, and several other studies have shown that there is increases in circulating ketone bodies during pregnancy, and we recapitulate that here, and because we also see increases in our transcriptomics and proteomics data set that BDH1, which is the main ketone body enzyme, is increased in expression in the maternal heart. We next inquired whether provision of the main source of ketone bodies, beta-hydroxybutyrate, its impact on maternal mitochondrial function. So what we see, interestingly, is that in response to beta-hydroxybutyrate, we see a significant increase in state-free respiration in mitochondria from post-birth hearts. We also see increases in state 4 respiration. However, this was not associated with any changes in RCR. So this leads us to the conclusion from our respirometry data that the maternal heart is able to utilize carbohydrate, amino acid, and ketone body sources for energy. However, it's most efficient when using carbohydrate and amino acid sources in comparison to ketone body sources. As I mentioned on the earlier slide, we also kept our remaining mitochondria for immunoblot assays. So we then went back and confirmed BDH1 expression in these isolated mitochondria and saw in support of our findings that mitochondrial BDH1 expression was significantly increased in post-birth mitochondria. In addition, with our other findings with respect to pyruvate utilization, we also see that there's a significant decrease in the expression of PDK4 in post-birth hearts. So knowing this, that circulating ketone bodies change during pregnancy, specifically at late pregnancy and beyond, and then also that we see increases in BDH1 expression, it's surprising that little is known regarding the key importance of ketone body metabolism in the maternal heart. Many studies have suggested that ketone body metabolism can contribute to metabolic remodeling in the heart, but this has often been associated with pathological insults, or specifically in the failing heart. So many studies utilizing a cardiomyocyte-specific BDH1 knockout mouse or overexpression mouse have provided insights with respect to ketone body metabolism in metabolic remodeling. However, little insight with regard to the metabolic remodeling that occurs in the maternal heart has been garnered. So that is the key direction of my currently funded R01 studies. And at the present time, we're specifically undertaking, at this very moment in time, in middle of study for this AIM2, where we specifically have generated a cardiomyocyte-specific inducible knockout of BDH1. But we're also in the early stages of the production of our liver-specific BDH1 knockout mice. So just a reminder that beta-hydroxybutyrate dehydrogenase 1 is a key enzyme in ketone body metabolism, converts beta-hydroxybutyrate into acetoacetate in the mitochondria. So we've generated this specific tamoxifen-inducible cardiomyocyte-specific BDH1 knockout mouse, and this is just some of the genotyping of BDH1 showing our wild-type heterozygous and floxed alleles. But we've also validated the knockout of BDH1 in the hearts of these animals. And I wish I would be in a better position to talk more about some of these studies, but I can't right now because we are actually in the final stages of our first study. But what I can allude to is that, because I'm blinded, I'm not entirely sure, but we at our post-birth time point, one of our genotypes is having significant issues with regard to their abilities to maintain a litter and also their ability, just their general health. So I hypothesize that that is potentially based on our data that we've accumulated with mitochondrial function with respect to the post-birth time point. I hypothesize that that is the knockout model. But hopefully this time next year, I'll be able to provide more of a comprehensive dive into those results. So what we've gone through so far is that during pregnancy, the heart increases in size, and this is associated with increases in transcripts associated with angiogenesis, cell cycle, and cell proliferation. And this is also associated with increases in transcript and protein expression of BDH1. By late pregnancy, the heart is further increased in size, and this is associated with changes in transcripts associated with metabolism. And we see increases in PDK4 expression, increases in BDH1 expression, and reductions in the phosphorylation of PFK2. By post-birth, we begin to not only see some of the reversal of the transcriptomic changes with respect to metabolism, but we also observe increases in extracellular matrix and fibroblast related transcripts. And we also observe increased mitochondrial respiration with carbohydrate, amino acid, and ketone body sources at this time point. And this is associated with a reduction in PDK4 expression and a sustained increase in BDH1 expression. We also see at these 2 time points, they're associated with the increased abundance and flux into pathways associated with cardiac anabolism and cardiac hypertrophy and pro-growth signaling, such that we see increases in metabolites associated with amino acids, glycerophospholipids, and nucleotides, in addition to ketone bodies. So hopefully I've shown so far that pregnancy-induced cardiac growth involves the coordination of several metabolic pathways, but as is with research, many unanswered questions remain. So one of those is, what is the contribution of liver-derived metabolites to pregnancy-induced cardiac remodeling? We already know that homoarginine and ketone bodies are produced in the liver, but we don't know the specific contribution they might play to cardiac remodeling during pregnancy, and even the potential roles within pathology. Our RNA-seq data set alludes to the fact that there are changes occurring in different cell compartments in the heart during pregnancy. So these need to be interrogated further, and also the organ-organ crosstalk which can occur, which can also impact pregnancy-induced cardiac remodeling. And then I've not really discussed it in too much detail, but establishing the full impact of the circulating hormones, the placenta, maternal-fetal interactions, and lactation on pregnancy-induced cardiac remodeling is important. And briefly, I do have pieces of data for these last 2 points that are upcoming that I can just quickly touch on. But establishing the nature of the hypertrophy that we see during pregnancy, and then also the impact of successive pregnancies on pregnancy-induced cardiac growth. So I'll first go to a piece of data alluding to the nature of the hypertrophy and then successive pregnancies. So what we had seen in our studies with our 4 time points, we had seen that the heart increases in size with respect to gravimetry, but when we assess cardiomyocyte cross-sectional area, we don't see a significant change at late pregnancy, where we see an increase in cardiac mass, but we do see an increase in cross-sectional area post-birth, which could indicate a switching from eccentric or a lengthening of myocytes to at late pregnancy, to then a concentric widening of cardiomyocytes at post-birth. And so one factor, there's several factors that could be at play in this hypertrophic response. It could be the myocyte lengthening. It could be increase in fluid in the heart, and it could also be, and probably very likely, but cardiomyocyte proliferation. So in order to try and tackle that a little bit, we took wet heart weights and dry heart weights to ascertain the fluid content of the hearts during pregnancy and post-birth. And what we observe here is that we don't observe any specific difference in fluid content in any of the hearts at any of the time points. And we see that this increase in heart weight, specifically at late pregnancy and post-birth, is maintained in going from the wet to dry heart weights. So that would be suggestive of the fact that we potentially ruled out any contribution of increased fluid accumulation. So we're now looking at cardiomyocyte length measurements in isolated cardiomyocytes. Next, I alluded to changes in the contribution of successive pregnancies to the maternal hypertrophic response. And so we had performed some pilot studies in which we had taken non-pregnant control mice and pregnant mice, and each pregnancy lactation cycle has its own non-pregnant control. But we did 1 pregnancy lactation cycle, 2 pregnancy lactation cycles, 3 and 4, and these mice were allowed to have I think it was a one week, or it could have been 1 to 2 weeks recovery in between pregnancies. But surprising to me is I had originally thought that with each subsequent pregnancy that maybe the heart would increase further in size, but it seems as though actually within our particular studies that with each subsequent pregnancy, the heart increases to a comparable size. And this is also mostly reflected with myocyte cross-sectional area. So this is actually good because we can actually use this as an indicator in some of our studies where we specifically interrogate pathology, because one of the pregnancy-associated pathologies that I'm most interested in, peripartum cardiomyopathy, actually occurs with successive pregnancies. You usually have to have two or three pregnancies to obtain that pathology. So having some baseline data there, knowing how the heart adapts to successive pregnancies under a normal condition is very useful. In addition to this, we have several additional studies undergoing right now. But I'd like to just now take the opportunity to not only thank my faculty mentors, but thank my lab and the internal collaborators that I have in the Center for Cardiometabolic Science. I'd also like to thank my external collaborators and R01 grant consultants for all the advice and assistance that they have provided. And I'd also like to thank the current and previous funding sources for which this work would not be possible. And then also my division and center for their support along the way. And with that, I'd like to thank you all for listening. I'd like to take any questions. Here's my contact. And then I'd like to leave you with today's event code so you can receive credit.
I'm so interested in what the liver is. So, and then I also think about ketone bodies and diabetes. So are there differences in outcomes for pregnant women and the heart that can be related to ketone bodies?
So I know, so typically during a normal pregnancy, they won't even examine ketone body levels. They'll only examine them during a diabetic pregnancy. So I don't think that they've made much traction with respect to like some of the underlying changes that occur there. I would expect that given some of the metabolic changes that would occur in the diabetic heart, some of that flux into those pathways responsible to growth would be perturbed. But that's something that we'd like to investigate. But you said you were interested in the liver, so
yeah, I think that is
the liver. So the heart definitely uses ketone bodies, we've seen, but I don't have data to suggest that it makes them. What we think is happening is there's a coordination between the liver and the heart during pregnancy. So that's why we specifically want to remove BDH1 in the liver to then target ketone body availability. But what is interesting, and I haven't showed it in this particular presentation, is that the arc of cardiac growth we see the same growth arc with the liver.
okay.
So I don't know if yeah, it could. I think it could be that, or there could be a additional circulating factor which could be promoting changes in the liver as well. We only of interest as well, we only really see changes in organ size in those organs that are associated with ketone body metabolism as well. So there could be some link there, and we've performed some stable isotope resolved metabolomics where we've actually interrogated multiple organ systems. So we've got the heart, the plasma, the liver, the kidney, and the muscle. So I'm hoping to be in a good position to show you those data next time. Well, if I'm not, I'll be very unhappy with myself. But
also very shocked when I saw the data about the maternal mortality. I think it came a few months ago and it was in the national news compared to, developing countries. So, and especially Kentucky and sort of the groups with the African Americans. So one comment in my customers, one is that I noticed more and more, like even today, the surprise pregnancies that we are seeing with the GLP-1s, where you tell the patient you may get pregnant, so better use some birth control or something. And many of them are using it either for type 2 diabetes or for obesity, weight management. So they already have a slightly higher risk. They've been, going into pregnancy. But they're not well prepared. So the pre-gestation clinics, which the MFM runs, where they prime the patient with the A1C, whether it's type 2 or type 1, then the BMI controlling the hypertension, and plus education of the patient, especially advanced pregnancies, where I had a patient who was in her 40s, never been pregnant before because she had severe PCOS.
Yeah.
Started GLP-1 and suddenly, though, and behold, at 8 weeks was pregnant. So, is that the maternal mortality more with advanced age or high risk pregnancies, or is it just healthy, normal people?
I believe from the studies that I quoted, they did break it down. And they did look at like the contribution of comorbidities, and I believe that I'm going from my memory, but I believe that it was either very close to or it was significant with respect to higher BMI, I think diabetes, and also age. Hypertension, most of those comorbidities. And then I think they also had a subset analysis where there was patients that had multiple factors and it was even more so. But most of the significantly alarming rates are actually in the Black population. So that's something that
the BMI and the hypertension. Oh,
yeah. So we need to work on reducing those rates and also increasing trust in the medical community, because a lot of I've read a few things about the Tuskegee trials, and a lot of the Black community are distrusting of the medical profession, and I don't blame them based on those studies. So instilling trust and providing stellar healthcare, I think. Yeah. And then also more women because they're entering pregnancy later, they've accumulated a lot of insults on their bodies just through being alive, exposure to environmental factors, lifestyle factors, which educating people on health and what is health and going into pregnancy healthy is also important.
So these animals develop pregnancy.
So the ones that we have studied are just the FEB or C57 Black 6J, and just a normal pregnancy, we don't see that. Usually the studies that have investigated those factors have either targeted a specific gene or process or given additional stressor. So like if you give so high fat diet. So we did some of those studies ourselves, and kind of curious because what we actually had seen during late pregnancy is high fat diet increased ejection fraction. So, we didn't see any indices of pathology, but I think we would see it with the multiple pregnancies and a stressor as well. So that's goals of the lab, to develop a physiological relevant model that recapitulates cardiovascular disease without necessarily targeting a gene. But we had originally believed that knocking out BDH1 in the heart would result in cardiomyopathy. It hasn't necessarily yet, but it's completely opened up a new area that I didn't even consider first off, but also that's just with one pregnancy. Maybe with multiple pregnancies, we will see a cardiomyopathy, but hopefully I'll be in a position to show you that next time.
Like, which ketone body does it use mainly for?
Oh,
Beta hydroxybutyrate or?
so the major ketone body that's used by the heart is beta hydroxybutyrate. There is also acetoacetate that it could use, but in our studies, in the mitochondrial ones specifically, we did try to provide acetoacetate, but we weren't able to get any mitochondrial respiration at any appreciable levels in response to acetoacetate. However, acetoacetate is never alone. It's always there's always beta hydroxybutyrate in the system when there's acetoacetate. So what we didn't do, unless I'm remembering incorrectly, is combine them in those studies. But yeah, the primary one is beta hydroxybutyrate. Yeah.
Years since I was involved in that. But might condition of pregnancy be considered stress? You know, kind of like the brain when it's under stress, just in pregnancy in general.
So
It can be kind of a stressful.
yeah.
Body made.
So I guess in the literature, they always refer to pregnancy as the nature's stress test for women. So there is a degree of stress because of the additional burden placed on the heart and such. We did, with respect to your comments on the brain, in our studies where we looked at the fluid content we also looked at different organs, and we did not see any changes in brain size at any time point during pregnancy, and we also didn't see any changes in fluid content there. So maybe in our hands it's not changing, but I know with respect to brain function, one of my colleagues at Mississippi is actively working on brain function during pregnancy, and they do see changes. They and I think with additional stresses imposed during pregnancy, they see adverse learning and memory outcomes. But I think the studies during pregnancy are still, unfortunately, in their infancy. Yeah.
And then, of course, PIEHS and Enviro Institute. So the idea of socioeconomic status and environment, environmental contaminants and heavy metals or other, to which certain populations may be exposed because of socioeconomic status and living, could play a role as well. Have you thought about trying to get like a little pilot grant from CIEHS to look at some kind of dual exposure model? Have people been doing that?
So I know in other institutes, some of my collaborators are looking at the environmental exposures during pregnancy. Also in the division here, Clara Sears, she's looking at the maternal fetal health in response to environmental exposures with like PFAS and but from an epidemiological standpoint. So we're actively collaborating. And then also I'm collaborating with Dr. Carl in the center who's doing e-cigarette exposures during pregnancy. So I think that what I would like to do is do different types of more physiologically and environmentally relevant exposures that would be applicable to everyone, yeah. So,
Shouldn't be one. But I think, the grandmother impacted the granddaughter.
Oh, yeah.
So I'm sure there's a, exposures going back generations, the zip code and.
the things that I am really interested in are the newer body of literature from the fetal cells going to the maternal heart and impacting that, and the physiology of the maternal heart, the microchimerism. That's kind of interesting, but I think it's probably beyond. And it could be why that there's differences with when you have a normal pregnancy versus a cardiovascular complicated pregnancy. Maybe there is that maternal fetal communication on that level too that's impacting everything. So
I mean, if the glucose gets too high, that's a problem.
yeah.
I don't know whether
So
the metabolism might switch somewhat to beta hydroxybutyric acid or something. I think the elimination more pregnancy. I have no
Yeah.
idea how much.
it does make more.
One of the things is that because there is fetal utilization and then the
Mhm.
glucose goes down, at that point you will make more.
Yeah.
So
And then
that's
increase.
one of the things that like in gestational diabetes, where they're not type 1, sugars are not really high, but one of the common things is they're not eating enough, because they're afraid the blood sugar goes up. So we tell them, we check their ketones, mainly to figure out if they're eating enough. Because what they will do is they will try to keep the blood sugar low. They don't want to go on insulin, so they don't eat enough.
Yeah.
And then you'll see the ketones go up.
Mhm.
And then we have to tell them, eat more,
Yeah.
eat carbohydrates, because otherwise they'll eat other things. And I think that won't help bring that ketones.
They have weight limitations, how much weight they can gain, and most of your gestation diabetics are higher BMI, and now they have weight cutoff, so if they take more carbs, they have to take more insulin.
Yeah,
You
that's
gain
the
more
thing.
weight.
Especially if they're not on insulin, they're always afraid that they will go
Go
on.
on.
And
So it's a very
Yeah.
difficult situation.
try to tell them not.
Starting off with a good BMI and blood pressure and good health,
Mhm.
that's, and not to have surprising. But
But in response to your question, during late pregnancy, there's a significant increase in circulating ketone bodies, which has been shown in humans and mammalian species across the board. But this is at a level that's physiological, so it's like between 1 and 3 millimolar, but it only is much more increased, for example, during like a diabetic pregnancy where it's like closer to like between like 10 and 15, no, sorry, 20 and 25 millimolar. And then you have exercise coming through the middle of those, where exercise is around 5 millimolar. So now it would be interesting to exercise pregnant mice, see what the implications are of that further increase in ketone body production. But we'll hopefully be in a good position. We've had a few struggles with our mice along the way.
Yeah, that's a nice work.
Okay, so typically both pregnancy and exercise usually induce physiological hypertrophy. They have some similarities, whereas we don't really get any detriments in function, we don't get any fetal gene expression or fibrosis, but some of the underlying signaling and metabolism contributing to both are different. Exercise is acute bout, or like a smaller time period, whereas pregnancy is 9 months. So, and then you have the hormonal changes on top of that as well. So there are differences, but there are similarities, and that's why they're often grouped together. Mm-hmm.
differently than other people that aren't active.
Yeah.
And I wonder if there were similarities in the biological chain. You know, pregnancy is only for a limited amount of time, and then it goes back, hopefully, to the way apart.
Yeah.
Maybe not so hopefully if you're better off, go out your eye.
Oh. So we have done those studies to try and like trace out specifically during pregnancy how long the increase in cardiac size is maintained. And what we see is it's maintained during the period of lactation. And then there's no difference there between the post-birth and post-birth non-lactation groups, but it's at least 6 weeks out. It's higher than a non-pregnant control. But we don't know at this time point when it goes back to normal. Our successive pregnancies data would suggest that it does go back fairly quickly, or it's just not long enough for it to go back and it just stayed bigger. That time period between cycles is too short, but it's something that we need to investigate. Oh, do I end?
Yeah, stop sharing.
No. Oh, there's a chat. Okay. Are there many publications on post-mortem studies of pregnant women who have died during and after pregnancy? I believe there are a couple, but they're very few and far between, and I think they don't consist of too many samples at present. We actively have a collaboration right now with the University of Kentucky, and we've actually received hearts from not only men and women with dilated cardiomyopathy, but we've also received hearts from women that have succumbed to peripartum cardiomyopathy. So we've started to look at some of the underlying biochemistry of those hearts. And we're seeing some metabolic changes, but we're at a stage where we're quite low powered to make any real insights. And then also the problem with our data set is there's a lot of differences within the clinical characteristics of the people providing the samples that we have. So I think with promoting awareness, and that will hopefully also promote additional inclusion of women into studies, pregnant women into studies, then we'll get some more insights into what's happening there. But I don't think there's that many postmortem studies that I'm aware of. So I think that was it. I can't see. It doesn't scroll any further. So
Okay.
okay, thank you very much.